can combine with the intercellular adhesion molecules and vascular cell adhesion molecules. Tumor necrosis factor and interleukin-1, they induce endothelial expression of ligands for integrins, mainly vascular cell adhesion molecules and intracellular adhesion molecules. On other side, these chemokines, they act on blood vessel by combining with proteoglycans. These are the proteoglycan molecules which are present on the endothelial cell wall. These chemokines get attached to this proteoglycan, and in turn, these chemokines act on adherent leukocytes. The change which is caused by these chemokines is integrins which are in low affinity state, they are converted into high affinity state by the reaction of chemokines with the leukocytes. And this integrin-mediated adhesion is a form in nature. After that, cell cannot move or roll on the endothelial cell surface, followed by transmigration of this particular cell through the endothelial cell wall. During the transmigration, another molecule is involved, that is platelet endothelial cell adhesion molecule 1 or CD31, which helps in transmigration of leukocytes through the cell wall. Transmigration, also known as diapedesis. You can have a short answer question on this. What is transmigration or diapedesis for 2 marks? Leukocyte migration through the endothelial cell wall. There are several adhesion molecules which are there present between the endothelial cells, but important is immunoglobulin superfamily member, that is PECAM1, platelet endothelial cell adhesion molecule, which helps in migration of leukocytes through the vessel wall. After the cell traverses the endothelium, leukocytes pierce the basement membrane. Outside the endothelial cell wall, there is basement membrane. This membrane is pierced by leukocytes by the secretion of enzyme collagenase, which will destroy the basement membrane, and that way leukocytes, they come to lie in the extravascular space. What is the importance of these adhesion molecules? Selectins, integrins, platelet endothelial cell adhesion molecule, VECAM, IACAM, is that person can have genetic deficiency of these adhesion molecules. When this deficiency is present, leukocytes cannot leave the circulation and reach to the site of inflammation or injury. That will lead to defective inflammatory process, and these persons are prone to develop recurrent bacterial infection. Once leukocytes come to lie in the extravascular tissue, that is after exiting the circulation, leukocytes move in the tissue toward the site of injury, and this movement is called chemotaxis, which is also defined as locomotion along the chemical gradient. Because what are the injurious agent, either microorganism, dead tissue, or debris, they will create or produce certain cytokines and chemicals, chemokines, and that will attract the leukocytes towards them. So that is locomotion along chemical gradient. What are these chemotactic stimuli which will cause chemotaxis of the leukocytes? Important chemotactic stimulus which is exogenous in nature and most common is bacterial products, peptides and lipids. Endogenous chemotactic stimuli are products of cytokines like chemokine family, particularly interleukin 8, components of complement system C5a, arachidonic acid metabolites. Significant member of this is leukotriene B4. So these are the exogenous as well as endogenous chemotactic stimuli. which will cause movement of the leukocytes towards the site of injury. What these chemotactic stimuli do? These chemotactic stimuli, they bind with the G protein coupled receptor. There are 7 transmembrane G protein coupled receptors. This chemotactic stimuli will combine with this, cause some cytoskeletal changes in the leukocytes. That way, cell will migrate towards the site of injury. The important changes which occur in the leukocytes subsequent to binding of chemotactic stimuli to this G protein coupled receptor is polymerization of actin filaments, which occurs at the one end of the cell with accumulation of myosin at the opposite end. So this is the leading edge of the cell where actin filaments gets polymerized. They will spread these filopodia, short or long thread-like processes, and cell is pulled by this filopodia toward the site of injury. The nature of leukocytes which infiltrate the site of injury or inflammation varies with the type of stimulus as well as age of the inflammatory response. You have to remember this. Like many of you may be knowing during your first year, they must have taught you, like in any acute inflammation, particularly bacterial inflammation, neutrophils are the predominant cell type in the initial phase. Say initial 6 to 24 hours, neutrophil will predominate. After 24 hours, 24 to 48 hours, neutrophils are replaced by macrophages or monocytes. But there are certain conditions where the nature of this infiltrate, leukocyte infiltrate differs. Like in Pseudomonas infection, there is continuous and ongoing recruitment of neutrophils at certain intervals. Whereas viral infection, lymphocytes are the predominant cell types which are present even from the beginning. In hypersensitivity reaction, you will have lymphocytes. Apart from that, macrophages and plasma cells. In some allergic reactions as well as in parasitic infection or infestation, you will have eosinophils as the predominant cell type present in the infiltrate. So nature of this infiltrate depends on the type of stimulus as well as the age of the inflammatory response. In any given infection or injury, neutrophils are the first cells to arrive at the site of injury. There are some certain reasons for that. One is neutrophils are more numerous in the blood than other leukocytes. During the differential count of blood, you must have seen neutrophils are about 70 to 75% in the blood. So that is one reason. Another reason is neutrophils respond more rapidly to the chemokines. Third thing is they get attached firmly to the adhesion molecules, which are rapidly induced on endothelial cells, such as P and E selectins. So these are the 3 reasons why, because of which neutrophils predominate in the initial phase of the inflammation. But after they enter the tissue, neutrophils are short-lived. They undergo apoptosis and disappear within 24 to 48 hours. On the other hand, monocytes or macrophages, not only they survive longer, but may proliferate in the tissues. And in a way, they become dominant cell population in the prolonged inflammatory process. Apart from inflammation, macrophages also play a role in the repair process. They produce certain growth factors and other mediators which help in the repair process. Once the cells have reached to the site of injury by the mechanism of chemotaxis, next step is phagocytosis. Attractant leukocytes will try to destroy the offending agent. Phagocytosis, there are again 3 steps. First, the attractant will recognize attached to the particle to be ingested. Once it gets attached to the particle, subsequently will be by that leukocyte. Following engulfment, phagocytic vacuole is created inside the leukocyte. This phagocytic vacuole gets fused with the lysosomal vacuole inside the cell and forms phagolysosome. That is the third step, killing or degradation of ingested material. So phagocytosis occurs in 3 steps: recognition and attachment of particle to be ingested, engulfment with subsequent formation of phagocytic vacuole, killing and degradation. So again, short answer question: what is phagocytosis? Write 3 stages of phagocytosis. There are phagocytic receptors present on the leukocytes, macrophage mannose receptors, macrophage scavenger receptors, and opsonins. These opsonins are immunoglobulin G, C3b, and mannose binding lectins. Once the particle gets opsonized, the process of phagocytosis becomes more potent. Opsonized particles can easily get phagocytosed by the leukocytes. This is a diagrammatic representation. Here is the phagocytic receptor which gets attached to the microorganism. Plasma membrane will spread out around this microorganism, will zip around this microorganism, will form phagosome with ingested microorganism. So microorganism is ingested in the phagosome. This phagosome, this is the lysosome. Phagosome combines with lysosome and forms phagolysosome. So within this phagolysosome, there are 3 processes which occur, which will destroy the microorganism or try to destroy the microorganism or ingested material. One is lysosomal enzymes will act on this microorganism, will try to destroy them. Another important thing is oxygen derived free radicals or reactive oxygen intermediates, which will also try to destroy this microorganism. And third mechanism is nitric oxide metabolites. So there are 3 mechanisms by which killing and degradation can occur: reactive oxygen intermediates or reactive oxygen species, reactive nitrogen species, and lysosomal enzymes. Intracellular killing of microbes and debris. The killing of microbes and destruction of ingested material is accompanied by reactive oxygen species, reactive nitrogen species, and lysosomal enzymes. Coming to reactive oxygen species, there is an enzyme complex called phagocyte oxidase, which contains at least 7 enzyme proteins. In a normal resting leukocyte, these different 7 enzyme proteins are distributed in different parts of the cells. Some of them are lying within the cytoplasm of the cell, some of them are lying in the cytoplasm cell membrane. But once the cell get activated, these enzyme complex gets synthesized at the phagolysosome. This is the phagolysosome. At the membrane of phagolysosome, this enzyme complex gets synthesized. This is phagocyte oxidase, also known as NADPH oxidase. The main function of this enzyme complex is it will cause oxidation of reduced NADPH, that is nicotine adenine dinucleotide phosphate. H is for reduced form. So this reduced NADPH is oxidized to NADP, and in the process, oxygen molecules is reduced to superoxide ion. Superoxide anion. This is O2 dash is superoxide anion, which is a reactive oxygen species. See, this superoxide anion by spontaneous dismutation is converted into hydrogen peroxide, which is again a microbicidal agent or bactericidal agent. But by itself, it is not that potent compared to other molecules, that is hypochlorite and hydroxyl ions. So these hydrogen peroxide in presence of myeloperoxidase enzymes which are present in the neutrophil granules. As you know, neutrophils contain myeloperoxidase enzyme, right? So this myeloperoxidase enzyme along with halide in the form of fluorine reacts with hydrogen peroxide and produces hypochlorite, OCl2 minus. Hypochlorite, which is a again active ingredient of household bleach that we use in the laboratories, important component of that. So hydrogen peroxide, hypochlorite and hydroxyl ion. Hydroxyl ion is also produced from the hydrogen peroxide. And here ion is required, Fe+2 is required. So superoxide anion, hypochlorite ion and hydroxyl ion, these three superoxide anions or reactive oxygen metabolites will try to destroy the offending agent. Right? These are the reactive oxygen species: superoxide anion, hydrogen peroxide, hypochlorite, and hydroxyl radical. So hydrogen peroxide, myeloperoxidase, and halide system is the most potent and efficient bactericidal system present in the neutrophil, which will destroy the offending agent. But the side effect of this is these oxygen-derived radicals may be released extracellularly from the leukocytes, and which may cause tissue damage that accompanies the inflammation. These oxygen-derived radicals can get released in the adjacent area and will damage normal tissue, which will exacerbate the inflammatory response or process. And to counter check these damaging effects of the oxygen-derived free radicals, there are certain antioxidant mechanisms which are present in the body in the form of superoxide dismutase, catalase, glutathione peroxidase, ceruloplasmin, and iron-free portion of transferrin. Third mechanism is nitric oxide derived free radicals. There are three different forms of nitric oxide synthetase: eNOS, nNOS, and iNOS. E is for endothelial nitric oxide synthase, which will synthesize nitric oxide in the endothelial cell wall. And the function here is this nitric oxide produced in the endothelial cells will maintain the tone of the blood vessels. N is neuronal nitric oxide synthase produced in the CNS, which helps in the neurotransmission. And I is inducible nitric oxide synthase, which is present or acts in the macrophages, will cause production of nitric oxide within the macrophages. These produce nitric oxide, then will react with the superoxide anion and will generate highly reactive free radical, which is known as peroxynitrite, ONOO-, peroxynitrite. So there are three mechanisms: lysosomal enzymes, reactive oxygen species, reactive nitrogen species. In the lysosomal enzymes within the macrophages and neutrophils, there are specific granules within the neutrophils. You can say like secondary granules and azuropheric or primary granules, which contains various enzymes, which will help in destruction of the offending agent. Important of these are acid proteases, neutral proteases, and again to check the excessive action of these enzymes, there are anti-protease mechanism present in the body in the form of alpha-1 antitrypsin, which will neutralize neutrophil elastase and will prevent further damage which is not required. Apart from that, there are also additional mechanisms for the destruction of offending agent, and one of them is neutrophil extracellular traps. These are extracellular fibrillar network. What they cause concentration of antimicrobial substances at the site of infection and will prevent the spread of microbes by trapping them in the fibrils. These neutrophil extracellular traps are the viscous network formed from the nuclear chromatin. Nucleus of the neutrophil gets exuded from the cell to outside, and it will form a meshwork of fibrils, which is viscous in nature. So will attract the antimicrobial substance, maintain their concentration, and prevent spread of infection in the distant areas, which is an additional mechanism of killing of microbes. One more mechanism is T helper 17 lymphocytes. As we have seen, phagocytic cells are neutrophil, neutrophils and macrophages. So these two are the important cells which play in the major role in the acute inflammation. But there are also lymphocytes which are part of acquired immunity, but still they play a role in the acute inflammation. The mechanism by which these T helper lymphocytes play a role is T helper 17 lymphocytes, they produce interleukin 17. And this interleukin 17 induce chemokine secretion, and these chemokines will attract other leukocytes and which will help in the process of inflammation. The significance of the T helper lymphocytes can be studied in persons where there is absence of this T helper lymphocyte response, and when this response is absent, these individuals are prone to develop frequent bacterial and fungal infections and also cold abscesses in the skin, where these cold abscesses will not have typical signs symptoms of inflammation in the form of redness and warmth. The part will not be warm to the touch and part will not become red. So these are known as cold abscesses, which are commonly seen in the skin. The person who is deficient in this T helper 17 lymphocyte function. So the main role of inflammation, as we have seen, is the removal of offending microorganism, maybe either bacterial, viral, parasitic, or fungus, as well as remove the damaged tissue. Like in a cause, if the cause of inflammation is tissue injury, there's some damaged tissue is there in the body or some part of the body. This has to be removed, and leukocytes play that role. But in the process, they can also cause damage to the normal adjacent tissue, which is known as collateral damage. And the classical example of this collateral damage is tuberculosis and some viral infection like hepatitis. So in the process of trying to remove the offending agent, like in tuberculosis, Mycobacterium tuberculosis, and viral infection, hepatitis viruses, the response is the exaggerated response, leukocyte response, which is produced, will cause damage to the surrounding tissue and further prolong the inflammatory process. Second is autoimmunity, where the cells fail to identify self antigen from non-self antigen and will mount immune reaction or immune response against the self antigen, that is known as autoimmunity, which is an inappropriate directed response. Third one is hypersensitivity, which is an exaggerated response. Any antigen which is like trivial in nature, like dandruff, dust particles, person can become allergic to that and will mount immune reaction or hypersensitivity reaction. Examples is asthma and drug allergies. So in these conditions, leukocyte mediated inflammation can be rather damaging to the tissue. There are other conditions where inflammation is a, you can say, beneficial response to foreign invaders and necrotic tissue, but may cause tissue damage. And these are the examples like type 2 diabetes mellitus, Alzheimer's disease, atherosclerosis, acute respiratory distress syndrome, rheumatoid arthritis, lung fibrosis, and certain types of cancers. Termination of acute inflammation. There are potent anti-inflammatory mechanisms present in the body. The main function of these anti-inflammatory mechanism is, once the function of leukocytes is over, these leukocytes are destroyed, and the mediators, chemical mediators which are produced in response to injury, they are again dissipated or neutralized. That way, inflammatory process is brought to the halt and further damage to the normal tissue is prevented. The important point here is there occurs switch in the type of arachidonic acid metabolites which are produced, which are also known as chemical mediators of inflammation, which are basis for this all the changes of inflammation, that is vascular changes and cellular changes. What happens here is arachidonic acid metabolites which are pro-inflammatory in nature, like leukotrienes, they become anti-inflammatory. Like initial phase of inflammation, pro-inflammatory metabolites are produced, but once the function is over, anti-inflammatory metabolites are produced in the form of lipoxin. So from the leukotrienes, production of leukotrienes, it is converted into production of lipoxygens, which are anti-inflammatory in nature. But still there are certain conditions where this mechanism also fails, and in those conditions, inflammation becomes chronic in nature. So this was all about acute inflammation. From the today's and yesterday's topic, you can have different full questions and short notes. Full question can be either in the form of like define inflammation, right? Vascular changes of inflammation and cellular changes of inflammation. So vascular changes here to write vasodilation, increased blood flow, increased vascular permeability, cellular changes, migration of leukocytes from within inside the cell wall, inside the circulation to the outside, followed by chemotaxis, phagocytosis, intracellular killing, right? Intracellular killing again mediated by three mechanisms: reactive oxygen species, reactive nitrogen species, and lysosomal enzymes. You can have different short answer questions, 2 marks from the topic. Like you can have what is chemotaxis, and write chemotactic stimuli. What is phagocytosis? Write 3 stages of phagocytosis. Okay. Write names of reactive oxygen species and antioxidant pheasants in the body. Short note: vascular changes of inflammation. Define inflammation and write vascular changes of inflammation. Write cardinal signs of inflammation and vascular changes of inflammation. Short note on the phagocytosis. Write a short note on phagocytosis. So you have to write 3 stages and explain in detail all the 3 stages. Thank you.